Hello, my name is Esther Duperchy. I am Director of Anti-Infective Research at Summit Therapeutics. I'm going to present data from a metagenomic analysis that evaluated the impact of the precision antibiotic Ridinilazole compared to Vancomycin on the gut resistome in a phase 2 study. As you know, the gut contains trillions of microorganisms. And because of the central role of this gut microbiome in human health, it is often referred to as a human organ. But it is also a potential reservoir of multi-drug resistant bacteria and their antibiotic resistant genes, which form the gut resistome. And we know that when the gut microbiome balance is affected by antibiotic treatment, it can lead to an infection of the colon by Clostridioides difficile. And C. difficile infection, or CDI in short, is commonly treated with relatively broad spectrum antibiotics that can further damage the gut microbiota. So the aim of this study was to evaluate the changes in the gut resistome of patients with C. difficile infection following therapy with vancomycin, a relatively broad-spectrum antibiotic, and therapy with ridinilazole, an investigational agent with targeted activity against C. difficile, and thus microbiome sparing properties compared to vancomycin. For this purpose, a microbiome analysis was performed on stool samples from 62 CDI patients who participated in a phase 2 study and were treated with 10-day oral ridinilazole or 10-day oral vancomycin. The fecal microbiota was analyzed by shotgun metagenomic sequencing, with sequencing all the genomes present in the samples. And thus it can determine the microbiome composition to the species level or lower, the microbiome function, and the microbiome resistance to antimicrobials. So I'm going to present you first the impact of antibiotic treatment on the gut microbiota and then the impact on the gut resistome. If we look first at the impact of antibiotic treatment on the gut microbiota at the phylum level, we can see that ridinilazole had minimal impact. In contrast, vancomycin treatment resulted in substantial changes with an approximate 16,000-fold reduction in relative abundance of bacteroidetes, 27-fold reduction in actinobacteria, and concomitantly a 5-fold expansion of proteobacteria. The driver for the expansion of proteobacteria at vancomycin end of treatment was a 21-fold increase in relative abundance of Enterobacteriaceae. This notably included an approximately 100-fold increase in Citrobacter and in Klebsiella species. As you know, these are important human pathogens that can carry multiple antibiotic-resistant genes. So after, we look at the changes in the gut resistome of the samples. In ridinilazole group, in green, the relative abundance of antimicrobial resistant genes did not increase at end of treatment. In vancomycin group, however, an increase in relative abundance of antimicrobial resistant genes was clearly observed at end of treatment. Looking now at the different classes of antimicrobial resistant genes, we observed that at the end of ridinazole treatment, none of the 21 classes detected in this study were significantly increased. In contrast, at the end of vancomycin treatment, 13 classes of antimicrobial resistant genes were in higher relative abundance. This included a 16-fold increase in carbapenem resistant genes, a 10-fold increase in polymyxin resistant genes, and an 8-fold increase in sulfonamide resistant genes. And these are common antibiotics used to treat Enterobacteriaceae infection. As you know, carbapenem and ESBL-resistant Enterobacteriaceae are classified as critical priority by the World Health Organization. So we looked into this and we observed that significant increase in the relative abundance of carbapenem resistant genes and in extended spectrum beta-lactamase encoding genes were only observed at end of vancomycin treatment compared to baseline. So, in conclusion, treatment with a microbiome sparing ridinilazole did not increase the gut resistome, which suggests that ridinilazole may have an additional benefit in the control of antimicrobial resistance. In contrast, at end of vancomycin treatment, we observed a profound impact on the gut microbiome and an expansion of the gut resistome. Expansion of the gut resistome included an increased relative abundance of genes conferring resistance to last-line antibiotics such as carbapenem and polymyxin. And this suggests that patients with CDI treated with vancomycin could potentially have a higher risk of serious subsequent infection. So I would like to thank the team at Clinical Macrobiomics for their collaboration on this project. And I want to remind that ridinilazole is an investigational antibiotic currently in clinical development for the treatment of CDI and is not approved by any regulatory body.